Hi, my name is Jayanti Mukherjee, and as a part of I Think Biology NPTEL course, today I am going to talk about my favorite cell. Let's get started. Do you recognize this picture? I am sure many of you do. So, what are these? These are something called stoma or stomata, right? You have heard of it. I'm sorry, the picture quality is not that good, but one of my students took it with her cell phone actually, and that is the reason the quality is a bit poor. But I'm a plant ecologist. I look at stomatal characteristics and behavior, and that is the foremost reason why my favorite cell are stomata. So, what are stomata? These are generally very microscopic pores, as you all know, enclosed by a pair of guard cells which flanks a stomatal aperture that that actually bows down or bows out when water fills in these guard cells. So we all know that, right? But I'm coming to that. That are found in the epidermal layers of the leaves and stems and also helps the plant to breathe. So stomata has multiple functions. We will go to that one by one. Let's take a look at where are they found. These are the basic structures of stomata, and this is how they look like when they are turgid and when they are flaccid. The first thing is when the stomatal pores are closed, as you can see water runs out, while when the stomatal pores are open, the guard cells are very turgid, and that's how it is possible to keep the pores open, right? So stomata are found in most land plants, right? Almost all of them, but the functions kept changing, and that is what we are going to talk about today and how it becomes very interesting to study stomata. So in lower dicots or eudicots, as you know, they are found mostly predominantly found in the on the lower surface, whereas in monocots they are found on both surfaces. Okay, and as you know that dicots and eudicots, what I mean are reticulate venation and broad leafed, whereas monocots are parallel venation and more thin needle type leaves, which you have been studying since you are in school, right? So there is no confusion there. So two broader shapes of stomata. This one, which is called as the, let me put the pointer out. These are the kidney shaped stomata and these are the dumbbell shaped stomata. Okay, so kidney shaped stomata are found on the dicots and eudicots, whereas the monocots have the dumbbell shaped stomata. So where and when this evolution took place and why, that is what is very interesting also to know. One of my undergraduate students and myself wrote this resonance article on stomata. Feel free to go and look it up, and these figures are all from that article. So stomata has shaped the evolutionary pathway of plants. Now why do I say that? So as you can see, these are the variations in structures of stomata, and we will go one by one, but look at what I have written here. Variation in structure and alignment of stomatal pore complex has been linked to the increase in efficiency of light capture and photosynthesis. Okay, so let's take a look at what are these. So first is the anomocytic stomata. You can see these ones. So these, you can see that there are, what is the difference between this and all the other on this list? These don't have something which is the yellow things, the yellow subsidiary cells. Those are called subsidiary cells. So these are the first initial types of stomata that evolved. Then slowly they started becoming little bit more complex. Two subsidiary cells which was horizontally placed, then vertically placed we have, then it divided into four subsidiary cells, and then of course we have this graminoid complex or the monocot dumbbell shaped stomata. which is two subsidiary cells which are very large, flanking the guard cells and the stomatal pores. Okay, and in some of the families also you see there are many more subsidiary cells, even two layers of subsidiary cells which has evolved. So these actually having subsidiary cells, the shape of the guard cell and the whole stomatal complex has made it over the evolutionary period more and more efficient in performing their role. Now, what are those roles? Here is what I mean. So did stomata evolve for photosynthesis? Let me ask you this question. Think about it. Were they always good at signaling this light and carbon dioxide? As in dicots and monocots, just as we looked? Actually not. Okay, so initially when stomata evolved, so early mosses are the first land plants that you will see. Before that, they were all grouped into protista. Algae, from where these mosses evolved, are called protista, are falls in the group protista. So early mosses had something like these on their sporangium, where the spores were found. So stomata on those sporangium had a sole purpose. And can you guess what that purpose could be? Was it gas exchange? No, it was actually, even though there could have been gas exchange, but the most important function of stomata at that time was desiccation, to helping in desiccation and dehiscence of the spores for spore dispersal. Okay, so when the sporangium was ready, the stomata will open up. It will dry up fully, the sporangium will be dried and it will burst and release the spores. So that was the initial function when it came. But of course, as you know, that once land plants started coming to land more and becoming independent, coming off the land surface, going to high as up as 100 meters, stomata became the most important cell that a plant could have. Okay, and as you all know, the main function was light capturing, light signaling, and also hormone sensing. So these were light capturing, and then there is another thing, we will come to that again. But stomata became very, very highly efficient in light capturing, better stomatal regulation, and increase in photosynthetic efficiency. This became, of course, over evolutionary time period, not in one day, right? And sensing ABA. ABA is, I'm sure you already know, it's called abscisic acid. It is a plant hormone, and it is called also the plant stress hormone. Whenever plant gets stressed, this is the first thing that is secreted, and roots start secreting abscisic acid. When it first senses its drought, ABA is transported to the leaves. Stomata immediately picks up that signal and closes. Okay, so here is drought there on the ground. We don't want to lose any more water. Let's shut down. So they are very good in hormonal signaling as well as carbon dioxide and light signaling. Most importantly, light signaling. Okay. But there are some evolutionary trade-offs or constraints that happen. What is that trade-off? One biggest trade-off is like if you compare stomata of plants with human, it is like if we open our mouth to take air or breathe, then we lose blood or we lose something very precious. And that's what happened with stomata. So when it's opening for photosynthesis, for taking carbon dioxide in, it was also losing water. So now this trade-off had to be balanced very much in order to survive in an environment. Because when the stomata is losing too much of water, the cell functions will stop. Okay, so cell can't carry on, it will get desiccated very fast. So stomata regulation has to be very efficient in order to pick up that signal. And that is giving rise to this carbon gain and water loss, thus gave rise to a multi-layer complexity in understanding this physiology and behavior of stomata. And there was plants actually tackled over the evolutionary time, you would be very surprised to know that they tackled this problem with one very small thing, which is the size density trade-off. There are other things too, but I am just going to talk about this. So initially when plant evolved, they had this kidney-shaped stomata, and the stomata in lower dicots were quite big in size. So what does that mean? If the stomata are big in size and they have to open up, so the minimum amount of water loss through those bigger pores were anyway much higher. So what happened was, even if they had fewer stomata at that time period, the water loss was much more. Then what happened during the early Cretaceous, late Cretaceous was the plants underwent some genetic changes. And one of those genetic changes was their genome started duplicating, and also it downsized. So these two genetic changes happened over the era. And what happened due to this was cell size and mechanics in the cell changed, and stomata size reduced. So in contrast to this kind of big stomata, you see stomata size now became very, very smaller. And this is one pattern of stomata, and this was found in, and where you could small, many stomata, whereas larger, only few stomata. So this is called the side density trade-off. Now why I am saying plants adjust? You will be very surprised to see, because as I said, if there are bigger pores, the minimum amount of water loss through these pores are more. So these kind of patterns generally came to see in more wetter climate. Whereas these other smaller stomata, whereas numerous stomata, are generally found. Actually, scientists are even now currently working on these things and finding that they are more associated with drier climates. And why? Because also stomata can behave in a very patchy manner. So it's not necessarily all the stomata at one point will be open. There could be few closed, there could be few open. It depends on where the light falls. Small specks of light can initiate the opening of stomata, and plants and the stomata on other parts of the leaf, same leaf, could actually remain closed to conserve water. Right? So this way, this size density trade-off also helped plants to adjust and generate more variation to adapt to varied climatic changes. Okay? So it's called patchy stomatal behavior. So now let's sum up. Crucial function. Why stomata? As you see, it's one of the most important cells on the plant and performs the most important job for us as well as the plant kingdom, which is photosynthesis, carbon sequestration, and as well as transpiration, which eventually also responsible for cooling our climate, right? So cooling our environment, cooling our climate. More recent research is showing that stomata has many more functions than just photosynthesis and transpiration. They are involved also in certain types of herbivory, certain types of pest control, and all those other functions. But the coolest are these two, and that is the reason why stomata are my favorite cells. Thank you for listening. I hope you enjoyed this video.
Hello, my name is Prachi, and today I'll be talking about my favorite cell. So before I talk about my favorite cell, I would like to start with the images of these two cells. These are the cells you might be familiar with from your textbooks, maybe from school or college. So look at these images. Can you guess which one is a plant cell and which one is an animal cell? I'm pretty sure looking at these images, you would be able to recognize it as a plant cell or an animal cell. As you can see in this image, on the right hand side, chloroplast is present, there is a central vacuole, and there is a cell wall. These all are the characteristics of a plant cell. And on the left hand side, you have an animal cell which does not have all these characteristics. So these are the cell pictures that we usually come across in our textbooks. But do all cells have similar structure? Do all cells have these same cell organelles? Look at this image. This image has three different cell types in it, but I want you to focus on the cells that I have marked here in black arrows. Maybe you can pause your video for a moment and take a moment to look at this image. Can you guess, is it a plant cell or is it an animal cell? Maybe not. These cells look nothing like the cells that we saw in the previous slide. So which cells are these? Let me tell you, these are the cells which are found in plants and they are known as vessel elements. And these are my favorite cells. And I'll tell you more about my favorite cells. Now, to begin with, you might have heard of the terms xylem and phloem tissue, right? You might know that xylem helps in the conduction of water and minerals in the plants, and phloem helps in conduction of food in plants. So pay attention that I'm saying the word tissue here, which means this tissue is made up of different types of cells. So let's talk about xylem tissue now. It is made up of four different types of cells: xylem parenchyma, xylem fibers, tracheids, and xylem vessels. And these xylem vessels, they are made up of different vessel elements, different cells joined together. Okay? And these are the main conducting part of the xylem. These vessel elements, these vessels, they help in the conduction of water and minerals in the plant. So let's learn more about these vessel elements. So just think about it for a moment. Here on the right hand side, I have a picture of one single cell, one single vessel element. How small is this? And these vessel elements, they join together and they help in conduction of water in the small trees and even in the larger trees that we see around us. So this image I have taken under a microscope, and this is the cell which helps in conduction of water in plants, in huge plants that we see around us. These are, as I told you, these are the water conducting cells and they are present in angiosperms only, which means they are present only in flowering plants. They are absent in pteridophytes and in gymnosperms. Some advanced gymnosperms may have vessels. Now the average diameter of a vessel element is usually 15 to 250 micrometers. Just pay attention, I'm using the word micrometer, and you may remember from your numbers and scales class in biology, we talked it in the second week. You learned about different units of measurement in biology, and micrometer, how small is that? So these vessel elements, they have such small diameter, it is in micrometers, and they help in conduction of water in plants. Another intriguing thing about them is that they are dead cells. They do not have any cell organelles. It just has cell wall around it, which makes it rigid and very long lasting. It is the lignin in xylem vessels which hold up the trees. Okay? So it has lignified cell walls. Now, as I told you, this is a vessel element. So as you can see in this image on the right hand side, there is a presence of perforation plate at the end of a single vessel element. So multiple vessel elements, they join end to form a single vessel. So as you can see on the left hand side, there is an image of a vessel. So as you see there, VE represents vessel element. So one vessel element is represented here with white dashed lines. So this is a single vessel element which joins with another vessel element at the end point. Okay, so multiple vessel elements are joined together to form a vessel, a water conducting tube. Okay, and these vessels, they are long tubular elements which run in vertical direction from soil to the ground. And this one single vessel that we're talking about, which is made up of different vessel elements, different cells, they can have as few as two vessel elements, or they can even have hundreds or even thousands of vessel elements. Okay, so now what do you think? Now I have told you that a single cell is known as vessel element. They join end to form vessel, and vessel helps in the conduction of water from root of the tree to the stem of the tree to the leaves of the tree. Okay, so what do you think? Is there a single vessel running from base of the tree to the very top of the tree? Is it a single vessel that is conducting water from the base to the topmost part? The answer is it's like a network or a mesh of vessels which help in the conduction of water. So now I want you to look at the image on the left hand side. So as you can see here, they have shown different vessels, and different vessels are joined side by side as well. As you can see, there is one vessel which is in light blue color. So as you can see, very faint dashed line is there. So this is one vessel, then there is next vessel, and then there is next vessel. So there are three vessel elements joined to form this vessel. Okay, but it ends here. It's not going to the very top. It ends here, but now it is connected to another vessel from the sideways, which is dark blue vessel. Now this vessel has two vessel elements, as you can see. One is here and one is here. So this light blue vessel is conducting water in the vertical direction from below to top, and then it is transferring the water laterally in the sideways direction to this dark blue vessel. And this dark blue vessel is further transferring the water to the red colored vessel here. And then it is transferring water to green colored vessel. So it's not like one vessel running from base of the tree to the very topmost of the tree. There is a network. Vessels end at certain point and then another vessels take up the job. So there is like a network sort of structure which runs throughout the trunk of the tree. Okay. So this is just a representative diagram, and the colors here are just to show you arrangement of different vessel elements within the tree. Okay. And the second image shows you the cross section. So if you cut a plant in a transverse direction, then you will be able to see different pores like structures, which are pores in a cross section. But we are mainly focusing on the vessels here running in the vertical direction. Okay. Moving to next, now in this image, so how are these vessels conducting water laterally? How are they transferring water to the next vessel that they come in contact with? So these vessels, on their walls, they have something called intervessel pits. Okay, so they transfer water from one vessel to another vessel through these pits. This here on the right hand side is a schematic diagram. Here you can see these are different vessels, and the circular structures, the small circular structures that you see here, they are intervessel pits. As you can see, the water is moving from this vessel in the upward direction, and then it moves to the sideward direction through intervessel pit. Okay, then it's going on the top, and then you can see these connections, right? So through these connections between different vessels, water can move laterally through these intervessel pits. So this mesh-like structure, it is all connected through these intervessel pits. Okay. So vessels, they're conducting water in plants in the vertical direction from base to the top of the tree, and they're also conducting water in lateral direction. So now, as I told you, these vessels, they do not have any cell organelles. They are dead cells. But was that always case? Were they differentiated like that? No. When these vessels are formed in a plant, they're formed from vascular cambium, which are meristematic cells. When they start forming vessels, the immature vessel elements, they do have cell organelles. They do have nucleus, they do have mitochondria, Golgi apparatus, endoplasmic reticulum, cytoplasm. But over the time, as they mature, they become dead. All the cell organelles, they disappear, and there is a secondary wall thickening. So I want you to think about it for a moment. The vessels are helping in conduction of water, right, as we talked about, and they do not have any cell organelles. So you understand their structure. These are tube-like structures with very rigid cell walls which do not have any cell organelles. So just think for a moment how their shape is reflecting their function. In the previous session, in your introduction to cells by Dr. Divya Uma, you already learned that how shape of a cell reflects its function. Now I want you to think for a moment how the shape of this vessel element is reflecting its function. So since there are no cell organelles, there is no obstruction in the conduction of water. So how the shape is reflecting function of this vessel element. Now We've talked about these vessel elements, their shape, their structure. But do they have same type of shape or same type of structure in all the plants? No. Even one cell type, it differs so much in different plant species. Okay, so different plant species may have different shapes of vessels. As we talked about their perforation plate, which is present at the end side of this vessel element, even they can be of different types. Okay, we talked about intervessel pits. Even their arrangement can be different in different plant species. We're talking about just one cell type, and even one cell type differs so much more in different plant species. Even this one cell type shows lots of variation in its shapes, its size, its structure. Another interesting fact about these vessel elements is that the vessel elements, they usually wider in the plants that are growing in warmer climates, and they are usually narrow in the plants that are growing in colder climates. Try and relate it to something like when there is like hot summer day, we get very thirsty and we need more water. Trees also need more water in hot and warm environments, right? So plants which are growing in warmer environment, they do have wider vessels to have more water conduction. Another interesting thing about these vessel elements is you might have heard of annual growth rings, which help in knowing the age of a tree. So in deciduous trees, usually found in temperate regions, they have ring porous wood. So in these type of trees, in deciduous trees, you will find two types of vessels which are formed within a year in one growth season. Okay, one will be early wood vessels and the other ones are late wood vessels. So these early wood vessels, they are distinctly larger than those which are formed in late wood. This ring porosity, it is considered as an adaptation to seasonal climate. Okay? The big vessels of early wood, they help in enhanced sap conduction, enhanced water conduction during beginning of the growth season. So just take a moment to think about what all did you learn about vessels and how cool are these vessel elements. So these vessel elements, they're dead cells. They have a very different structure than what you are used to of seeing in your textbooks. These small tiny vessels, they help in the conduction of water in the large trees that we see around us. Even in different plant species, they have different type of shape, size, structure, and even in the same tree, you can find different types of vessels. As you can see in this picture right here, you can have two different sizes of vessel within the same tree, right? Usually in deciduous tree, and these two different sizes of vessels are distinctly different. To summarize, in the previous lecture, you learned about three favorite cells from three different instructors. You learned about Toxoplasma gondii, diatoms, and Mixotricha paradoxa. And in this lecture, you learned about stomata and vessel elements. So, what is the difference between these two lectures? Just take a moment and think. In the previous lectures, all the cell types we talked about were unicellular organisms, single cell, which is in itself an organism. And in today's lecture, we talked about two different types of cells, which are cell types found in plants, right? So take a moment and appreciate the cells which make up our living world. We say that cell is the fundamental unit of life. And this fundamental unit of life, it differs so much. It has so much of variation. And how amazing is that? So I want you to remember that the cell type that we usually see in our textbooks, cells are much more than that. They have so much variation in their shape, their size, their function, and I would like you to go and search for your own favorite cell. And with this, we'll end today's lecture. And I hope you enjoyed learning about all of our favorite cells. And I hope you can appreciate that how many different cell types are out there. Thank you. See you in the next lecture.